Alright, now let's move back to investigations. Specification number 3 asks us to investigate, using a suitable stain, the pathway of water in a cut stem. This experiment involves the use of different dyes, as you can see here, so let's take a closer look. And of course, before we begin any practical, we need to talk about safety. So, here are the key precautions for this investigation. To avoid staining your hands with food coloring or dyes, it's best to wear gloves during the experiment. If you're working with concentrated dyes or chemicals, always wear safety glasses to protect your eyes from accidental splashes. Make sure the workspace is well ventilated, especially if the dyes have strong odors or could cause irritation. Never ingest food dyes or chemicals, and ensure all beakers and containers are clearly labeled and kept away from food and drinks. Keep all dyes out of reach of children and pets. And finally, when cutting the celery stems, use scissors or a sharp knife with care, cutting in a controlled manner to avoid injury. Because, safety first, always. This is the setup we will use to investigate how water moves in plants. We are going to work with celery stems and different colored food dyes so that the movement becomes easier to see. First, we will get our materials ready. We need fresh celery stems, plain water, and a range of food dyes in red, blue, green, and yellow. Now, here is what we will do. We will place several celery stems in separate containers. One container will have plain water, which will act as our control, and the others will each have water mixed with one of the colored dyes. We will then leave these containers in sunlight for a few hours so the celery stems can absorb the water and transport it through their tissues. After enough time has passed, we will cut cross-sections of the celery stalks using a scalpel on a white tile. This is where we will observe the results. We will look closely at the slices to find the vascular tissue, especially the xylem vessels. If the dye has been taken up, the xylem will be stained in the color of the solution we used. Without the dye, the xylem is hard to identify, but with it, we can clearly see the pathway of water movement inside the plant. So, we will follow this procedure to investigate water movement in plants. We will start by taking 5 celery stems of equal size and length so that our experiment stays uniform. Next, we will prepare 5 separate beakers. One beaker will contain plain water without any color, while the other 4 will each have water mixed with a different food dye, red, blue, green, and yellow. We will then place each celery stem into one of the beakers, making sure the freshly cut end of each stem is fully submerged in its solution. After that, we will leave the beakers in a sunny spot for a certain period of time so the water can move up through the plant. It is important to keep the celery in a location where the temperature and light remain consistent. This way, external factors will not interfere with our results. Once the time has passed, we will examine the stems and leaves to look for any color changes. The presence of dye inside the xylem vessels will show us the pathway of water movement. Using different food dyes will make it easier to track this movement, as the colored water will travel up the stem and reach the leaves. This setup not only helps us see how water moves through the xylem but also allows us to compare how each colored solution behaves as it travels through the plant's vascular system. After the set period of time, we could see that both the plain water and the dyed water had been absorbed by the celery and traveled through the xylem to reach the leaves. The celery stems placed in plain water showed no visible change, their stems and leaves remained the natural color. However, the celery stems in the colored solutions told a different story. The stems in red, blue, green, and yellow dye each displayed clear signs of coloration. The red and blue dyes created distinct, bold streaks running through the stems and into the leaves. The green and yellow dyes also colored the xylem and leaves in their respective shades, making the movement of water through the plant easy to trace. In every case, the most intense color was seen near the base of the stem, right where the dye had first entered. From there, the color gradually faded as it traveled upward. When we cut a cross-section of the stems, we could clearly see the colored regions marking the location of the xylem vessels, the exact pathways that carried the water and dye. Finally, the leaves of the celery stems in colored solutions showed the most vivid shades at their edges. This is where the water and dye had been transported all the way to the outer tissues, completing their journey through the plant. From these observations, we can conclude that the presence of color in certain areas of the stem clearly marks the pathway of water movement, revealing the exact location of the xylem vessels. The experiment demonstrated that the food coloring was absorbed by the celery and transported all the way through the xylem to the leaves. The celery stem placed in plain water showed no color change at all, while the stems in the colored solutions, red, blue, green, and yellow, displayed distinct colored streaks in both the stem and the leaves. These streaks are clear evidence of dyed water moving through the plant's vascular system. When we examined the cross-sections of the colored stems, we could see the dye concentrated within the xylem vessels. This final confirmation leaves no doubt about the path water takes as it travels through the plant from the roots or stem base to the leaves. In conclusion, this investigation clearly demonstrated how water moves through plants using food coloring as a visual aid. By placing celery stems in water mixed with dye, we were able to watch the colored water travel through the xylem vessels, tracing its path from the base of the stem right up to the leaves. The results confirm that the xylem vessels are the structures responsible for transporting water throughout the plant. The use of food coloring made this process easy to see, providing clear and vivid evidence of the upward movement of water and highlighting the vital role of the xylem in plant transport. So, this is a simple yet highly effective experiment that clearly demonstrates the movement of water in plants. Using food coloring not only makes the process visible but also engages learners by providing a striking visual result. The method is easy to carry out with basic materials, making it ideal for classrooms while still producing accurate, scientific evidence of how xylem vessels function. So, that wraps up the first unit of Chapter 7, Transport in Flowering Plants. I hope you found it clear and helpful. Don't forget to check out the notes I've linked in the description, they'll really help you revise. and I'll be back soon with the next part of this chapter. Until then, take care and keep learning. Stay tuned.